Now, in eukaryotes, there are some additional processing events that occur to the messenger RNA before it is considered complete. The three processing events include a mechanism known as splicing, and splicing is what you see in the top panel of this figure. With splicing, we need to take one step back and talk about eukaryotic genes and sequences overall. In eukaryotes, the gene itself is composed of intervening blocks known as exons and introns. The exons are the ones that contain important information for the synthesis of a protein, and the introns do not. Eukaryotic genes are referred to as mosaics in this sense because they have these intervening blocks of exons and introns. So in the splicing mechanism, what has to happen is the components, the introns, that essentially contain no information need to be removed, and the exons need to be pasted back together again. And this is because we just discussed that transcription starts at the beginning, at the transcription start site, and ends at the terminus. So if you have these intervening blocks in the DNA sequence, they will also be copied into the RNA sequence, and you can see that in the center of this slide. The darker yellow indicating the exon sequence, and the lighter yellow indicating the intron sequence. So splicing is actually a very complex and highly regulated mechanism. Essentially what happens is a cut is made on either side of an intron. The intron is removed, and the two ends of, in this case, let's say exon 1 and exon 2, are spliced or pasted back together again. And you see the same thing happening with the removal of intron 2. A cut is made on either side of intron 2 so that the end of exon 2 and the beginning of exon 3 can be pasted back together again. And this is referred to as a fully spliced mRNA. In addition to splicing, there are two additional processing steps that occur with eukaryotic messages. That is the addition of a structure known as the 5' cap, and then at the opposite end of the transcript is what's known as a poly-A tail. Both the cap and the tail are added on to the transcript after transcription. They are post-transcriptional additions. Both serve to protect the RNA from degradation by enzymes known as RNases. By having a structure at the 5' end, it makes the 5' end unrecognizable to these enzymes that could degrade the RNA. At the 3' end, we have the addition of a poly-A tail, which is a stretch of A-containing nucleotides repeated over and over and over again. The average length is about 250 A-containing nucleotides. This too is thought to protect the messenger RNA from RNases, because as the enzymes would begin to degrade the RNA, it would begin degrading the A-containing nucleotides and not the important information. Both the cap and the tail do play an important role in protein synthesis, which we'll see in our next lecture. Okay, so let's go ahead and look at these processes in a little more detail. We'll go ahead and start with the 5' cap addition. As I mentioned previously, the addition of the 5' cap provides many functions or serves many functions for the messenger RNA in a eukaryotic cell. One function that the cap provides is an element of protection. By adding the 5' cap or a modified nucleotide to the 5' end of the messenger RNA, this makes it unrecognizable to nucleases that would potentially degrade the RNA. So by having this modified cap structure at the 5' end, again, nucleases find the messenger RNA unrecognizable and are unable to begin degrading the messenger RNA. In addition, the cap plays a role in translation or protein synthesis, and we'll review that or come back to that when we cover the lecture that deals specifically with translation or protein synthesis. So the addition of the cap involves a multi-step process. As you see on this particular slide, there are 3 different enzymes involved with the mechanism of adding the 5' cap. What we'll do is we'll go through them step by step, and hopefully you'll get a good understanding of the mechanism involved in the addition of the cap. So the first step that you see in panel A at the very top of the figure is representation of the 5' end of the messenger RNA. The first enzyme that comes into play is an enzyme known as RNA triphosphatase. This particular enzyme removes one of the phosphate groups from that very end or terminal nucleotide at the 5' end. It's important to note at this particular point that the very first nucleotide is in fact a triphosphate nucleotide, meaning the 3 phosphates are present at the very 5' end of a messenger RNA. And that's what's depicted in this figure with the 3 P's indicating the phosphate groups. So the first enzyme in panel A removes the terminal or the end phosphate, so that what you have is a messenger RNA with the very 5' end only containing 2 phosphates. The next step in the process utilizes an enzyme known as guanylyl transferase. This is what you see in panel B. In panel B, you see the addition of a G-containing nucleotide to the 5' end, and it is this enzyme that is adding that G-containing nucleotide. So, hopefully you'll note at this particular point that every single cap is a G-containing nucleotide, and that holds true for all messenger RNAs. The next enzyme to play a role in this process plays a role twice in steps C and in step D. This enzyme is known as methyl transferase, and as its name implies, transfers a methyl group to specific locations on this cap structure. The first one in panel C shows you the addition of the methyl group to the nitrogen in position 7 of the cap guanine. So that guanine that was added on, or the guanine containing nucleotide that was added on in step B or panel B, that specific guanine gets a methyl group added to the number 7 nitrogen in the guanine structure. In step D, this enzyme also adds a methyl group to that very first nucleotide, and in this particular case, you can see it in panel E, all the way in the far right corner is a methyl group highlighted in red. That's the addition that's occurring in step D or panel D. So again, methyltransferase adds a methyl group to 2 different positions in a 2-step process. One methyl group gets added to the guanine that has been added on as part of the cap structure, and one methyl group gets added to the very first nucleotide in the messenger RNA. Now, one more thing that I want to point out is that even though we talk about these post-transcriptional modifications that occur sort of after transcription, this particular mechanism actually occurs very early on in transcription. Usually the transcript is about 30 nucleotides in length when the cap addition occurs. This makes a lot of sense because the idea here is to protect that messenger RNA as soon as possible. So almost immediately after transcription begins, that cap structure is added on to add that layer of protection so that nucleases don't start degrading the messenger RNA before the completion of transcription. This is really just another figure to highlight that cap structure. Shows you a little more of the molecular detail for the cap structure. The nucleotides in purple or highlighted in purple are part of the messenger RNA, and the one that is highlighted in the pinkish color is the G-containing nucleotide that has been added on as part of the cap structure. And I like to highlight this particular figure with looking at the molecular structure so that you can see a little more detail of the addition of that structure at the 5' end. What hopefully you can see from this is that that nucleotide is actually added on in the opposite orientation from typical nucleotides in the messenger RNA. And it is this addition in the opposite orientation that then makes the 5' end unrecognizable to those nucleases that we talked about that would be potentially degrading the messenger RNA. So now let's move on to the addition of the poly-A tail or the polyadenylation mechanism. In this particular case, this is a slide we saw in the previous lecture, and I'm not going to go over the step-by-step process of polyadenylation here. We talked about it in significant amount of detail as it is tied or coupled to transcription termination. So if you recall, you can go back to that lecture where we looked at the step-by-step process of the addition of the poly-A tail and then how that is followed by the termination of transcription. But I just want to highlight that this is, again, a post-transcriptional modification that does in fact occur, and it's lumped together with these processing events. So I wanted to refresh your memories by putting this slide in. But again, I'm not going to go over the detail here. You can go back to the previous lecture and look at the detail for the polyadenylation mechanism. However, I do want to remind you that polyadenylation also serves the purpose of protecting the messenger RNA, because by adding these A-containing nucleotides to the very end of the message, it protects the message from degradation. Now, it doesn't truly protect it from degradation. If enzymes begin to degrade the messenger RNA at the 3' end, because there is now this significant length of A-containing nucleotides, it's those A-containing nucleotides that would be degraded and not any important information. In addition, the poly-A tail does play a role in protein synthesis or translation, and just like with the cap, we'll come back to that concept when we talk about protein synthesis. Okay, so I've already mentioned that both the cap and the tail add a layer of protection to the messenger RNA, basically protecting it from degrading enzymes or RNases that would be in the cell, potentially degrading the RNA. I also mentioned that it plays a role in what I'm referring to as translatability. Having a cap and a tail definitely plays a role in allowing for the messenger RNA to be translated more efficiently. We're going to review that topic here in just a minute, and again, we'll come back to it when we get to protein synthesis and translation. In addition, I have not yet mentioned that both the addition of the cap and tail play a role in transport mechanisms. In eukaryotes, transcription occurs within the nucleus of the cell where the DNA is housed. The messenger RNA then must leave the nucleus, enter the cytoplasm, and encounter or engage with ribosomes in order for protein synthesis to occur. So transport of messenger RNAs through the nuclear membrane into the cytoplasm is a really important mechanism, and it turns out that having a cap and tail does play a role in this transport process. In addition, cap and tail components can play a role in more efficient splicing. We introduced splicing at the beginning of the lecture, and we're going to talk a little bit more about splicing in just a minute. But the cap and tail do play a role in helping with efficient splicing mechanisms as well. So I want to talk a little bit more about how having a cap and a tail on a messenger RNA helps with what I'm referring to again as translatability. So we'll be referring to the data in this particular table in just a minute. What I want to highlight is how this particular experiment was conducted and some of the details associated with it, and then we'll jump into looking at the data overall. The first thing that I want to mention is that messenger RNAs have what's known as a half-life, And a half-life is really just described as the amount of time the messenger RNA is around in the cell. More specifically, the definition of the half-life is the time it takes for half of a given RNA to be degraded. So this is something that can be measured, and there are messenger RNAs that are much more stable and are around in a cell for a lot longer period of time. And then there are some messenger RNAs that are much less stable and get degraded rather rapidly for one reason or another. The mean half-life or average half-life of messenger RNAs is somewhere in the ballpark of about 20 minutes. But obviously that's just a mean, and there are some components or examples with much shorter half-lives, as well as examples of much longer half-lives. So in the particular experiment that I'm going to discuss associated with this table, what the researchers did was look at the half-life of a specific mRNA. The mRNA that they utilized codes for an enzyme known as luciferase. Luciferase is most commonly found in fireflies, and it is what allows for bioluminescence in the fireflies, basically the fire in the firefly's tail. The light that's produced is by oxidation of a pigment called luciferin, but the enzyme is the one that catalyzes this reaction and allows for the bioluminescence to be detected. Okay, so in this experiment, they utilized luciferase mRNA, the mRNA that codes for that particular enzyme, and modified a number of features or characteristics of that messenger RNA. So in the very first column of this table, what you can see is the messenger RNA and the features or characteristics of that messenger RNA for each assay that they conducted. So for example, there were uncapped messenger RNAs that also did not have a poly A tail, indicated as poly A minus, or an uncapped messenger RNA that did have a poly A tail, noted as poly A plus. Conversely, they had capped messenger RNA that did not have a poly A tail, poly A minus. And lastly, capped messenger RNA that did have a poly A tail, poly A plus. So you have every combination of cap and tail. So the very next column looks at luciferase half-life in this particular experiment. And what you can see is that there's not really much difference in the half-life with any of the combinations, except for having a capped and a poly A-tailed mRNA. You can see that there is a significant increase in the half-life when both a cap and a tail is present. In the next column, what the researchers looked at was the luciferase activity. So now this is specifically, so by measuring the luciferase activity, this gives some indication of the amount of enzyme that's actually being produced in these cells. So in the first case, uncapped with no poly A tail gives a certain amount of luciferase activity. And then you can see uncapped with the addition of a poly A tail almost doubles the amount of luciferase activity. However, what you can see when you jump to a capped message that does not have a tail, there is a dramatic increase in luciferase activity. And then lastly, capped with a poly A tail sort of goes through the roof overall with luciferase activity. What this indicates or suggests is that having both a cap and a tail is really important for getting these messages translated overall. And again, we will come back to that when we get to protein synthesis or translation. So we just completed talking about the addition of the 5' cap and the addition of the 3' poly A tail. Both of these are considered non-template additions, meaning that they are not coded for in the gene sequence and that they are added post-transcriptionally. They are not coded for in the template, again, non-template additions. In the very first slide of this lecture, I gave a really good introduction to what's happening in the splicing mechanism. So just to reiterate, most eukaryotic genes are considered mosaics, meaning that they have alternating blocks of sequences known as exons and introns. The term exon is actually short for expressed codons, EXONS, and introns is a condensed form of interrupting codons, INT, and then ONS. Exons, just to help you remember, are components that will exit the nucleus of the cell. This is my mnemonic device. And introns are going to be components that stay in the nucleus in the splicing process. Okay, so the summary that I mentioned in the first slide, I'm going to go over really briefly again, in that what has to happen in the splicing mechanism is that, first of all, transcription of the gene needs to occur, and you get what's referred to as the primary transcript. The primary transcript may be referred to as pre-mRNA, immature mRNA, or hnRNA, which is short for heteronuclear RNA. All of these are examples of the description of the transcript that results immediately after transcription. The best term, in my opinion, is immature RNA, because it describes that this is a mRNA that needs to have additional things happen to it before it exits the nucleus of the cell. So a quick summary of what happens during splicing is that you have your primary transcript where the intron is recognized, specifically the exon-intron boundary, and then the corresponding intron-exon boundary. So either end of the intron is recognized. Cuts are made, and that piece known as the intron is popped out, and the two exons are then pasted together or spliced together. This is, in fact, a really complex mechanism. There are multiple ways that splicing can occur in eukaryotes. There are mechanisms that occur spontaneously. These are known as group 1 and group 2 introns, and they occur spontaneously, meaning there's no additional help that allows for the splicing to take place. Group 3 introns, which we'll get to on the next slide, are going to be the most common type of introns, and those do in fact require help from additional components. Again, we'll get to that in just a minute. One last thing that I want to mention, that for any given gene, the number of exons and introns and the position of those exons and introns is always the same. Differences occur when you compare gene to gene to gene. So gene 1 to gene 2 to gene 3, for example. They differ in the number of introns that you can find. They can also differ where the position of those introns are. So again, for a given gene, intron-exon position is always the same, and the number of introns and exons is always the same. However, when you compare one gene to another, that's where differences can occur. For example, there have been genes that have been identified that have no introns in them whatsoever. And there are some that have numerous amount of introns, all the way up to 362 introns. That's an awful lot of splicing that has to occur for that particular gene. So as I just mentioned, there are 3 types or categories of introns. Group 1 and Group 2 are considered self-splicing, meaning they don't need the assistance or the help of any other components in order for splicing to occur. And then Group 3 introns, which is probably the most common, is referred to as spliceosome-mediated introns. This is splicing that requires the help of a very complex molecular component known as the spliceosome. The spliceosome is a structure that is made up of numerous proteins as well as RNA molecules. So again, it is a very complex macromolecular structure that plays a role in allowing for the splicing to take place. Now, we're not going to talk about every individual component of the spliceosome. We're just going to consider the spliceosome as one entity that is, again, playing a role in mediating the splicing event. But it is the spliceosome that recognizes exon-intron boundaries in order to make a cut, as well as intron-exon boundaries to make the second cut, and then to bring the exon ends together so that splicing can actually take place. And that's what you're seeing in this really complex figure. Again, we're not going to look at each individual component, but I want to make sure that you're aware that it is a complex mechanism overall. The one thing that I do want to highlight is that it turns out that there's really not a lot of sequence complexity in recognizing both intron-exon boundaries or exon-intron boundaries. And there are some very simple consensus sequences that have been identified, but it's not nearly as complex as you would think it would be for recognition of those boundaries between an intron and an exon. In fact, about 60% of genes have some sequence elements that are recognized, but these sequence elements, again, are only found in about 60% of the genes. So that still indicates that there's a significant amount that doesn't follow the consensus sequence rule, even though spliceosomes are recognizing specific boundaries where to make cuts and where to paste things together. The last thing I want to highlight is that there are numerous diseases or medical afflictions that are associated with splicing errors. So lupus, which is an autoimmune disorder, is one example. And in this particular case, there is a problem with splicing. And the problem occurs because in individuals that have lupus, oftentimes have a situation where their own spliceosome components are recognized by their immune systems as something foreign, and antibodies are made against spliceosome components, targeting spliceosomes for degradation or destruction. And you can imagine that if spliceosomes are targeted for destruction, that could cause lots of different problems overall in an individual that is afflicted with this particular type of autoimmune disease. Another example is something known as Duchenne's muscular dystrophy. Duchenne's muscular dystrophy occurs in about 1 in 3,500 male births. The gene associated with this disorder is located on the X chromosome. So just to give a little bit of backstory to understand this particular type of muscular dystrophy, we're going to talk about a specific protein that is known as dystrophin. And dystrophin is a protein that interacts within muscle cells with an integral membrane component of the muscle cell, as well as the actin inside of the muscle cell. And this is what allows for the structural integrity of the muscle cell. So dystrophin sort of acts as an anchor between the cell membrane and the cytoskeletal components inside the cell. So if that protein is not formed properly, then you have cells that don't have that rigid structure associated with them. So it turns out that the gene that codes for the dystrophin protein is a rather large gene. It's large mostly because it contains lots of introns. There are 60 exons and 60 introns in this particular gene. So 60 different splicing events have to occur in order to produce the properly spliced dystrophin messenger RNA, and then that would result in the proper production of the dystrophin protein. So individuals with this particular type of muscular dystrophy oftentimes have splicing errors that take place because, again, there's so many splicing events that have to occur before you get a mature dystrophin messenger RNA. And when these splicing events don't occur properly, the end result is a protein product that is not formed properly, hence the muscular dystrophy that we just talked about.